Hello everyone, welcome. I apologize for the long break. I had been away and doing some research this summer and was unable to post any content the last couple of months. But we will be posting more in the coming weeks, and we'll be back on our regular schedule of posting at least a video a week, if not more. So before the break, the last video that I posted was in chapter 10. It was about talking about the structure and function of genes and how we as molecular biologists or cellular biologists study these structures and function. How do we determine what a gene may look like? How do we determine what its function might be and what it might be useful for? So today we are going to continue on that journey by focusing this particular lecture on function of a gene. So previously we have already talked about how we might isolate and clone DNA molecules, how we might sequence a gene of interest, a particular DNA fragment of interest, and look for possible genes in there, look for possible functional products in there, whether they will be a messenger RNA at the end of the day or a gene with a protein that codes for a protein that may have some important function. We've talked about how we can use PCR to utilize PCR, polymerase chain reaction, to create clones of that important or interesting fragment of DNA, and then clone it in other cells, animals, or structures so that we can examine it further and explore its function. So today we're going to be focusing all about the functionality of these gene products. We're going to do that by first looking at the mRNA, which is that intermediate in some case and the final product in others. So that mRNA is the first step usually that we examine because it provides a snapshot of all the genes that might be expressed in a particular system under a particular condition, under a particular developmental stage, whatnot. mRNA is essentially all the messenger RNAs that are present in a particular cell or in a particular organism at that time that you are collecting that specimen. And we can do that through various mechanisms. We'll talk about a few of them in a few minutes. But one of the ways that we do that is through in situ hybridization, which can reveal not only when a gene is being expressed, but also where it is expressed. So in situ hybridization is a really, really cool technique where even if we do not know the protein product that it makes yet, but we have that messenger RNA and we have that sequence, we can use a method to essentially hybridize it to the messenger RNA inside that body structure or on a specimen that we have already collected and sectioned, and then we can look at exactly where that messenger RNA is present and being expressed essentially. So here is an example of how it can help us detect its existence and its expression. So it can be used obviously to detect a particular messenger RNA being expressed anywhere in any tissue, in any part of the body, but it can also be used to detect presence of just viruses, for example, a viral RNA that is present inside the cell. All you need is a probe with some kind of a fluorescent or colorimetric marker tag linked to it so that you can find it and see where it is. And that probe essentially comes in the form of the messenger RNA complementary sequence, a small tag. It doesn't have to be throughout that mRNA. It's usually just a small tiny tag made of anywhere from 20 to 50 nucleotides, and it's tagged with usually a fluorescent probe so you can visualize it. So in this case, in this micrograph, for example, we have cultured epithelial cells. These were infected with HPV or human papillomaviruses, and then they are stained, and essentially stained, first of all, for nuclei with the blue parts in here. Those are DAPI stained. So they are fluorescing in one color, and then the cytoplasm has a green fluorescent stain to it, right? So you are able to see it, see the body of the cells individually, and then you have these red probes. The red probes, or the pink probes that you see, or the pink dots that you see, are the viral mRNA sequences that are being attached by the probe that has a fluorescent tag on it, so that you can see and visualize where exactly it is. So the viral virus-infected cells, they have this particular probe indicating that the viral particles, in these cases, the viral probe is binding to the viral particles inside the nucleus. So this would be essentially the viral mRNA or viral RNA that is inside these cells. You can also use the same method to locate genes on isolated chromosomes as well. So let's say you have a new sequence. You don't know where it came from because you maybe do not have the entire sequence of that particular animal yet, and you want to know where exactly this this might be found in all your chromosomes in that particular case. Well, in that case, you can make a tiny probe complementary to those sequences that you are interested in locating. Again, the probe is made by taking the complementary sequence attached to a fluorescent tag usually, or a colorimetric tag, and then you use methods to hybridize it inside the cell, inside the isolated chromosomes, for example, and you look at where exactly it binds. And looking at where it binds, you can locate not only which chromosome it is on, but also which arm of the chromosome is it, and how many copies are present throughout the genome. If it is a sequence that may be a sequence like, let's say, DNA binding domain, for example, it may light up a bunch of places inside your genome, and you will know that this is not, this is a repetitive sequence of some sort that may have regulatory function or may have some other important function that you should be aware of. So in this case, you have isolated chromosomes that you can see, a pair of chromosomes. This is human chromosome 5, taken from a mitotic cell in metaphase. And usually that's where we want to take these chromosomes, right, when they're in chromosomal state. So it's going to be during mitosis. The DNA probes that are being used are for different sequences, and they are each tagged with a different type of fluorescent tag. So each one is going to have its own color that it's going to line up with. And here you have both the paternal and the maternal copies lined up next to each other. So you can look at how these copies are doing. So in this case, if you notice, you have 4 essentially dots showing up for each one of the probes. That's because they are undergoing mitosis at that time. They've already replicated their chromosomes, so you have the 4 copies right now. And you have them aligned side by side, in this case, maternal and paternal copies of the chromosomes, so you can see all 4 copies of each one of those sequences. This technique is also nicknamed fish fluorescence in situ hybridization, and that's what it is called. So another way that we can look at some of these gene expression to determine where and when this might be expressing is by using a reporter gene construct. In this case, you are going to have your normal gene sequence, so this is, again, this is going to be a little bit of genetic engineering involved in here to create a reporter gene construct. So for you have your sequence here, you have your normal genes right here, and then you have the starting site for the RNA synthesis, and you have the coding sequence for a particular protein of interest. And this particular protein of interest then is going to be expressed in certain conditions. We can replace the coding sequence of gene X by putting in a reporter gene, let's say GFP, right, green fluorescence protein, and you still keep your regulatory DNA sequences that determine this expression pattern and create a new recombinant reporter gene construct, which has the regulatory sequence of your original coding sequence that you are interested in. But now you don't have the coding sequence for protein that you wanted, but instead have a reporter gene in there. Like I said, it could be green fluorescence protein. And now you will be able to see exactly where these regulatory sequences cause the expression of that gene to occur, because that's when and where the green fluorescence protein will light up. So maybe they only get expressed during the, early embryonic stage in the liver cells or in the newly developing neurons. And then you will see them light up just in those cells at those times and at no other times. So you can see where your protein of interest is going to be expressed and when it's going to be expressed by using this type of a construct. You could also do that using the in situ hybridization here that we talked about, but this would be more cell biology way of looking at it, where you would want to then take samples from all the different organs or all the different parts at different stages of life at different times and examine them by probing them separately, and that would become a lot more involved project than if you were to just use a reporter sequence and do a more genetic tailored approach. So these can determine, obviously reporter genes can really help us understand how genes are regulated, gene expressions are regulated, and what each type of regulatory sequence is responsible for. Because if you remember, in this case, in the normal gene, we had 3 different regulatory sequences. So you could create multiple reporter gene constructs, each, one which has all 3 sequences together, one that just has, some that just have 1 of them in each. So just 1, just 2, just 3, or just 1 and 2, just 1 and 3. And then examine exactly which ones are switched on, when just one of them is present, which part or which space does it express our gene. So here we have these 6 different spaces that we are looking at and 6 different types of cells that we are examining. And we see that regulatory sequence 3 only leads to expression of cells and expression of this protein in one type of cells, which is the cell type B, let's say the neurons. However, regulatory sequence 2 turns on the expression of these cells in 3 different types of cells. Maybe it's the epithelial cells as well as the, the liver cells, or it's like site-specific or type-specific that it's switching it on. And then when we have just one present, it actually turns it off and not on. So you are unable to see its expression by itself. And so by looking at all the sequences together, you are able to determine what exactly it's able to do. So if you looked at just one by itself, you saw nothing was expressed. If you looked at 1 and 2 combined, you saw, well, you still saw in 2 different types, but now one of them was gone. So then you know that one is responsible. If one is present, it's not going to allow it to be expressed in certain type of cells, but the others can still be active. So that again is a really important way that we use reporter gene constructs, and this is a very genetic approach, genetic engineering approach. Going more to the cellular side of things, again, you can use the green fluorescent protein tags, or there are actually lots and lots of fluorescent tags that we can utilize nowadays, YFP, RFP, others as well. And they can be used to identify specific cells in living animals, again, by utilizing this tag to that particular type, some protein that is expressed only in that type of cells. And then you're able to see that network and see exactly where those types of cells are present and what exactly is their biology. We can use the GFP that fluoresces at different wavelength like I said, so that you can make the connections between different types of cells and see how those are interconnected, how they might be connecting with each other, how they might be interacting with each other, or working separately from each other. All of these would require a little bit of genetics and a little bit of cellular biology approaches. So here in this example, we are using multiple GFPs that fluoresce at different wavelengths to reveal the different connections that particular individual neurons have inside the brain. And so you can see how that network of neurons is prepared in that particular cell type. Now, once we know where these cells are functioning, we usually want to know exactly what do they do where they are. To do that, the standard method to find out what it does is to knock it out. What happens if it's not there? What goes away? Is it able to survive? Is it, so is it like lethal altogether? Is it embryonic lethal, or does it let our organism develop to the full size, but then it's unable to survive? Is it hindering a function that is essential or non-essential? Is it only affecting certain population under certain conditions? Maybe they are not able to regulate their temperature in heat, or maybe they're unable to utilize a particular amino acid now in their diet. So looking at what exactly goes away helps you figure out what it does. The standard of, again, examining this has been RNA interference, or RNAi, which is where we inhibit specific genes by using interference of RNA interference. In this case, you take a small, tiny piece of that particular gene transcript from that mRNA. You create a double-stranded RNA out of it. That double-stranded RNA, anytime our cells see double-stranded RNA, the automatic response of that cell is to think that there is too much of that messenger RNA present in the system. And by default, they will just degrade all the messenger RNA that is remaining in that system at that time, thinking that it is no longer needed. And that causes an inhibition of those specific genes. Sometimes it's complete, where all of it is removed. Sometimes it's just a reduction, 25%, 100%, 75%. And that reduction can show you how it is functioning. Now, if you have small organisms or easy to genetically manipulate organisms, for example, E. coli and C. elegans are really, really easy to manipulate in this case. You can just feed them this double-stranded RNA, and they can just knock out completely the function in the particular gene, of that particular gene that you're feeding them the double-stranded RNA for, and the embryos that are developed from them are also going to have the knockouts of this as well. In this case, you can very, very easily see the effect, a systemic effect of these particular genes. You can also do it in cell culture, where you take cells that are already in culture and then transfect them with this RNA interference product and with this double-stranded RNA, and then examine its effect in vitro. You can also do it just in the liver or just in the brain if it is something that is going to be lethal if it is everywhere, and examine what it does when it's just affecting the muscles or just affecting the livers or just affecting the brain, and then examine it that way. However, again, the first method is usually to examine it at a more organismal level, or either at that level or at a more just cell culture in vitro level. In this case, you have E. coli that are expressing this double-stranded RNA. So we transfected in the E. coli, so they had it, they're producing it, they're essentially acting as little factories. And you feed these E. coli to the worms eat it up. As they eat it up, they are now going to have this double-stranded RNA in them. this is going to reduce the effect of that particular gene in all the cells, all the tissues, including their reproductive organs. So all the embryos that form in there are going to have that particular gene silenced. So not only are you going to be examining the effect in the adult organism, but also you will be looking at how it affects their reproductive ability and how it affects the resulting progeny from this. So here in panel B versus C, in panel B you have a wild type worm embryo. So from these C. elegans, where you see the egg and the sperm pronuclei coming together by cell fertilization. They are going to be forming the fully fertilized embryo soon. However, in C, you have the particular gene silenced, and in this case, these nuclei fail to migrate and join together and merge together. So this is extremely important. So this would obviously cause this particular worm to become sterile in this case, because those can never get together. They can never form the function. They can never form a viable organism that can have any function. So this lets you know that that has some important function in embryonic development at most likely, or even the fertilization process. So again, going forward with that, obviously the gene can be deleted. The gene can be inhibited, as we saw here. We can also just delete the gene altogether or replace it with a different version. That's another level of construction that can be done to explore its function. So one of the ways that we do that is through targeted gene replacement. A lot of times in mice, but again can be done in C. elegans, in yeast, in different cells. So in this case, you have embryonic stem cells that are growing in culture. We take the altered version or the targeted version of the gene that we have created and introduce this fragment into the cells and allow them to proliferate. We take the cells from the colonies that have the DNA fragment in them. So these we test for. We test each one of the colonies. We make sure that they have that particular gene replaced in them. So this one is going to have one copy of the target gene replaced by an altered gene. But remember, that still means they still have one normal copy. The other one is messed up. This is going to be then injected into an early embryo that is formed. So we isolate early embryos from pregnant mice, and then we inject these cells, these embryonic stem cells, with one copy of the target gene, altered or replaced, and we inject this into an early embryo. This is going to lead to some part of the mouse having the mutation or the altered form of the gene, while the remaining is going to remain the same way. And these cells, once they have formed, gone into those embryonic stem cells, once they're part of that early embryo, are introduced back, usually not to the same mouse, but into pseudo-pregnant mice and allowed to give birth. Now, some of these offsprings are going to have those germline mutations in them with the altered gene that can be tracked in some way or another. This type of mouse, but obviously these are going to have genes in different places, right? It's not all going to be the same. Some of them are going to be just in their somatic cells, but in some cases, these are going to have that mutation incorporated into their germline. And when it is incorporated in the germline and you mate them, you are able to get mice that have both copies that are altered. So here, what they have done is they have mated them with normal mice, and the offsprings are going to have just males and females, which will have one copy that is altered and one copy that is not. And then when those two are mated together, you will have some mice. If you remember back, if you have two mice that are both heterozygous for that mutation or that alteration, you can have 25% of the progeny have completely altered form. And so here you're going to end up with transgenic mice in which both copies of the target gene are altered. These type of mice are, obviously this is going to take some time, right? Because it takes several months for an animal to become, for mice to become adult, where they can have their own progeny, where they can be mated. So this takes a couple of years usually to get good mice. These type of mice are called knock-in mice, right? So when you have these mice where cells have the altered genes in all of their cells, they are going to be knock-in mice. Now here is an example of this. These are transgenic mice with mutant DNA helicase. Here in B, A is a normal mouse, and these mice show the effect of that mutated DNA helicase by showing premature aging. So helicase gene is involved both in transcription, which is the common part of it that we know about. It is also part of DNA repair, obviously, because then again it needs to work to unravel the DNA so that it can be utilized and changed and copied. So compared with a wild-type mouse, which you see here, when you have the defective gene, you have a lot of those premature aging. They have osteoporosis. They look pretty skinny and sad, right? They look like they're not fed well, even though they're getting the same amount of nutrients, but they are unable to utilize them properly. They have early graying of their hair, infertility, and reduced lifespan. So obviously this particular gene is extremely important, and it mimics human mutation that causes that is characterized by brittle bone syndrome, brittle hair, skeletal abnormalities, and reduced life expectancy. And so this allows us to examine the effect of this particular important gene in a setting where we can fully understand its function and fully unravel what exactly it does that causes those effects. Now, we can do that. That's great, but sometimes it's not going to be so simple, right? So in this case, you were still able to get a mouse that was viable, that even though it has reduced lifespan, but it did have infertility. So we couldn't continue to get more progeny from it and understand it further for multiple generations. Sometimes you're unable to get anything out of it because it's an embryonic lethal, and it's unable to give you a viable organism to begin with. In this case, what we need to do is what we call a conditional knockout, where a gene can be selectively disabled or disrupted in just targeted tissues. So let's say we do have an inkling of what this particular gene might do. Maybe it affects, it is known to induce liver hypertrophy or liver cancer or liver cirrhosis. Or maybe it's something that we already know that it's mutated in a lot of muscular dystrophy patients. So we can create what we can call conditional knockouts, where it is only going to be expressed in that particular organ at a time when we create the conditions to switch it on. One of those systems is what we call a Cre-Lox system. This requires insertion of two separate engineered segments of DNA, so two separate constructs, so that already makes it more complicated. Each one with particular controls on them. So in the, and this needs to go directly into the germline cells of that animal so that you can create the organisms from it that have the progeny has the particular engineered version of the gene. So we want, because again, it is going to be a more complicated, more step process, we want to make it very targeted, very clean, so that that's the only copy that the animal has. They don't have the normal one under their typical control, and they are able to examine its effect properly. So in this case, you have one construct that encodes a recombinase, which is usually the Cre recombinase. This Cre recombinase is under a liver-specific promoter because we are wanting to create the final construct only active in the liver. If it was supposed to be under, let's say a neural in the brain, then we will do a neural-specific promoter, or any other organ system, we would want to do that specific promoter. And then in the second construct, we are going to have our gene of interest under, that's going to be on, usually with these two LOXP sites that are flanking it. The LOXP recombination sites are what are going to be recognized by the Cre recombinase, and that's what's going to help it excise out and be deleted from that particular target tissue. So in this case, once you put these two constructs inside the particular animal cells, inside the particular organism in the target tissue, the liver-specific promoter will make sure that it only gets activated inside the liver. When it's active, it will turn on the Cre recombinase gene, which will make Cre recombinase. Now, on the other side, the gene of interest was always on until the Cre recombinase binds to the LOXP sites, excises it out, and now you have an altered chromosome where there is no gene of interest present. And that gives you that final construct, the final version, where the animal does not have that particular gene expressed in that particular target tissue. So this is extremely useful. This can be used to examine, again, targeted function of a particular gene of interest in a in the organ of interest, or in the organ that we think it has the most important function in, without disrupting it everywhere and creating a lethal construct. A newer method that is, that has been on in the last few years, in the last decade, is the Crispr system. This is extremely precise. So this is extremely elegant and precise. And so this has become kind of the new gold standard coming behind, in front of CreLox in coming times. In this particular system, it is using a bacterial system. It's a very sophisticated, elegant system used by bacteria to edit their genes. This is called CRISPR. You have your target gene that you want to remove. So CRISPR is present in archaea and bacteria. It's usually because of certain bacteriophages that were absorbed by the bacteria, and it stands for clustered regulatory interspace short palindromic repeat sequences. These are targeted by this Cas9 protein, which is an enzyme essentially that can utilize these a little bit like the LOXP sites and cleave sequences within those CRISPR sites. So in this case, you have your target sequence, you have the cleavage site that is going to be recognized by the Cas9 to cleave it, and this is going to be leading to your altered version of targeted gene that is going to be introduced through homologous recombination in this site where you have the double strand break caused by Cas9 protein. So in this case, when you have certain conditions in place, the Cas9 protein, when it is inactive with the transcription activator, it's going to lead to the gene being on. When you have the catalytically and inactive Cas9 fused with transcriptional repressor, it's going to lead to the targeted gene being off. And so this can be used both to regulate the expression of that gene, but also to replace your original gene sequence with a target, with the altered version that you want to utilize to look at maybe the effect of the mutated version of that gene or the normal version of the gene, depending on the condition. So in this case, the Cas9 protein, which is again enzyme, is going to guide the RNA design that we designed by based on what type of binding site it has, so that it can bind to that sequence in the target gene and allow for that homologous recombination to happen once that targeted break is made. So one of them is going to be obviously associated with the Cas9 protein, and the other end of this guide RNA is going to bind to the complementary sequence on the target gene, allowing for the cleavage to happen and for the recombination to happen. So here, when we introduce it into the cell, we are basically introducing the Cas9 protein with this 3' end of the guide RNA already binding to the Cas9, and the cleavage, the guide RNA is then going to bind to the target gene at the cleavage site. Once the double strand break is made, the altered version of the target gene is introduced in there. This is with the target gene that the altered version that you have produced, and that is going to be then combined in there through homologous recombination, leading to your replaced gene version. Now, this promotes a very quick as well as precise conversion of that gene into the one that you normally look at. Cas9 can be also utilized, like I said, to activate a normally dormant gene or to turn off something that's already always on, and then examine what effect it has at the end of the day. Now, a lot of those were looking at targeting gene expression in different animals, different organisms. In plants, you can also use some additional techniques, and transgenic plants can also be made using recombinant DNA techniques. These are techniques that are optimized for plants specifically. So one of those is by taking discs of leaf and incubating them in a culture of agrobacterium that has the genetically engineered construct that you want to create, that you want to utilize. In this case, the selection medium that we grow the plant's discs in will only allow the cells that have acquired the DNA from these agrobacterium that had that genetically engineered construct, so that those are the only ones that are going to start to grow. Once they have started to grow a shoot, so first we start to create a callus, and then you add the shoot-inducing medium, have the plants start to form, and then you allow them to grow in another medium where the roots can form, and then you can grow those seedlings into appropriate plants. These involve, so this involves several steps of carefully manipulated and controlled growth factors and mediums that can have each part of the process happen at the right time. This type of technology is used all the time in agriculture to create what are sometimes introduced as evil genetically modified organisms and genetically modified plants and food items. Not all of them are evil. Some of them have no issues, no problems, but they are used, for example, some of them are used to produce rice grains that have really high levels of beta carotene or other proteins and other vitamins, essential vitamins and nutrients, so that they can be provided to populations where maybe there is a limited amount of resource, limited amount of foods available, and those people are able to get the appropriate nutrients from a smaller amount of food. So in this case, there were two different enzymes that were needed to be altered. So you had the phytoin enzyme from maize and a lycopene enzyme from bacterium that are incorporated to create this beta carotene rice that has higher levels of beta carotene in them. Now, when we want to have large amounts of proteins produced by these different constructs that we are making, we utilize simpler cells or simpler model systems like yeast, bacteria, or cells grown in culture, where we can essentially utilize them as factories for us. Any expression vector that we are able to transfect or introduce into them with our own sequence that we are interested in, will be massively produced inside those cells, and that overexpressed mRNA will lead to overexpressed protein. You can, purify this protein and then use this purified protein in assays to examine its effect on cells, its effect in different enzymatic reactions, or to understand its process. And that's a great power of being able to do genetic manipulation like this. These recombinant DNA techniques obviously help us understand what each protein does, what each gene functions for, and it allows us to make understand them at a better level. So here we have, for example, a gene or cDNA of interest. We can insert it into a gene expression vector, put it inside a host cell, whether it is a mammalian cell or a yeast or a bacterial cell. We can overexpress it, purify the protein, and now once we have the protein, we can determine its sequence. We can determine its different peptides that can be formed with it. We can search for DNA database for gene sequences. And we can also utilize it in enzymatic assays to examine what its possible function might be. And all these things help us understand its function at a cellular and an organismal level. So just kind of reviewing all the stuff related to today's lecture, we examined how messenger RNA can provide a snapshot of gene expression in gene expression profiling, RNA sequencing studies. We can utilize this further by using techniques like in situ hybridization to reveal when and where a particular gene is expressed. We can also use reporter gene constructs to allow for specific proteins to be tracked in living cells to see when they are expressed, where do they go, what do they do, what do they interact with. We can also use these reporter gene constructs to examine regulatory gene sequences and see what each one of their functions might be. Then we can also delete or, introduce or delete different mutated versions of the genes to help examine what the gene function is, how it affects it. One of the ways that we can do that is through RNA interference, which inhibits the activity of specific gene. We can also delete or replace them entirely with altered versions, deleted versions with known mutations, using Crispr or Crispr system, both at a cellular or organismal level. Genes are edited and examined with great precision using the newer Crispr Cas9 system. That's the one that is coming out to be the new gold standard for these things. So if you have any questions, please put them in the comments below, and I will make sure to answer them on the site as well as in a new lecture if needed. I will be doing another lecture that was asked from the viewers over the break. I think it was on the sexual history or that particular chapter, sexual reproduction. I will do that next, and then after that we are going to be continuing with our genetic series as well. Any other things that you would like to see, please do send me a note, and I will make sure to bring them on in the coming lectures as well. I will be posting some lab demos and newer lab techniques in the coming weeks as well, as I introduce them in my own classroom as well. So keep a lookout for those too. Hope you enjoyed. Take care.